Good morning, everyone. Welcome to Leading Breakthroughs, Gene Drives for a Sustainable Agriculture and Biodiversity Conservation. This webinar is part of a series organized by ISA Inc. in collaboration with the DE Biotech Program Office. I am Janine Escasura from ISA, and I will serve as the moderator for today's event. Recent advancements in biotechnology has been in the fields of genetic engineering, gene editing, and gene drive. This webinar series intends to provide discussions on the new and emerging agri-biotechnology tools and innovations. The first of the series provided information on the recent developments in the applications of gene editing in crops and animals. Today, we will delve into the science and application of gene drives in food, agriculture, health, and biodiversity. As of now, we have more than 80 participants joining us today. Please be reminded that this webinar will run for 2 hours and will be recorded for documentation and evaluation purposes. So, before we start the webinar, please allow me to quickly remind you of the house rules. First, Zoom attendees are requested to keep their microphones on mute unless invited to speak. We highly encourage our audience to engage and actively participate during the Q&A session. If you have any questions, you may post your questions using the Zoom Q&A button seen at the bottom of your screens. Please do not forget to indicate to whom you are directing your question. These questions will be addressed during the Q&A session. Lastly, please complete and fill out the online evaluation form to receive your certificate. A link and QR code will be flashed at the end of the webinar, and only those who have accomplished the post-webinar survey will be given the e-certificate. We also have a set of activity objectives that we would like to achieve at the end of the webinar. First, this webinar intends to provide information about the science and application of gene drives in food and agriculture. This webinar also aims to provide information about gene drive applications to address invasive species and protect biodiversity. This webinar also highlights the state and rationale of ongoing research in this field. Lastly, this webinar intends to be a venue for stakeholders to clarify issues and concerns about the technology. To formally start the webinar, may we call on the Executive Director of ISA Inc., Dr. Rodora Romero Aldemita. Dr. Aldemita is a multi-awarded scientist and science communicator. She holds a PhD in botany from Purdue University, USA, and a postdoctoral fellowship on golden rice at Albert Ludwigs University, Germany. She coordinates the activities of ISA's Biotechnology Information Centers, as well as capacity-building activities on biotechnology and biosafety in Asia in partnership with USDA and Philippine DA Biotech Program Office. She is also a member of the Technical Advisory Team of the Applied Biotechnology Committee of DAPPO, a board member of the Biotechnology Coalition of the Philippines, the editor-in-chief of Philippine Journal of Crop Science, and the scientific reviewer of 6 scientific journals. Everyone, please welcome Dr. Rodora Romero Aldemita.
Wow, that was a big applause for Janine for opening our webinar today. Thank you so much, Janine, for the opening salvo, something like, hey, we're here. And although it's a bit a very fast introduction, I hope that our participants were able to get what we really would like to have on today's webinar. So currently, I'm looking at the number of participants, and they're still coming in. So we're around 101. The registrants were supposed to be how many? 292, and we hope to achieve 50% of the number of registrants. So to open, thank you so much again, Janine, for that wonderful introduction and for opening our webinar today. So good morning, good day, everyone, or good evening, everyone, wherever you are in this part of the world. Welcome to the ISA webinar series titled Leading Breakthroughs, Gene Drives for a Sustainable Agriculture and Biodiversity Conservation. We already know the different biosciences, biotechnologies, and gene drive is something new still for some of our audiences. So we are the International Service for the Acquisition of Agri-Biotech Applications, a nonprofit organization based at the International Rice Research Institute, Los Baños, Laguna. We envision a world where accurate biotech information empowers farmers and consumers to drive food security amid so many agricultural challenges through various innovations, and that includes GeneDrive. Our mission is to feed the world with knowledge on biotechnology by supporting transformative technologies. What are those transformative technologies? So these are innovations that will help us sustain our environment and at the same time ensure food security, fostering impactful partnerships, and facilitating science-based policies toward food secure communities. So this webinar, as what Janine said, is part of the webinar series in collaboration with the Philippine Agriculture and Fisheries Biotechnology Program on an array of topics about new and emerging technologies in biotechnology in the global and Philippine context. We had the first webinar on gene editing, and these were on crops and animals. And this second webinar is now on gene drives. So gene drive, as I said, is one of the innovative strategies designed to spread specific traits in a population of organisms. This technology offers transformative applications for food, agriculture, health, and biodiversity. So in today's webinar, we will feature experts who will present to us and expound in detail on the science and application of gene drives in food and agriculture and their application to address invasive species and biodiversity conservation. During their presentations, they will also highlight the state and rationale of ongoing research in their field. At the end of their presentations, there will be a Q&A portion, and this will serve as a venue to raise questions on the technology and its applications. And I hope you will take this chance to raise all your concerns, your queries, your doubts, so that you can be clarified, of course, and that you can share this learnings to your peers, to your colleagues, to your students, so that we hope that by the end of this 2-hour webinar, our participants, especially our young and aspiring scientists, will have learned about the technology, its application, and you can share this again with your peers. So we also invite you to visit our ISA website. At our website, we have a resource on gene drives and other biotechnologies which are written in simple language and downloadable for free. So I hope that you can avail of all these resources to help you out in understanding gene drives and all the other biosciences and biotechnology. Finally, we are glad for the attendees' enthusiasm. We are now, how many are we right now, who are attending this event? So we are now 132. And we will start the ball rolling. Again, we hope that this 2-hour webinar will enhance your understanding of gene drives. Happy learning, everyone, and have a good day. Back to you, Janine. Thank you.
Thank you very much po, Ma'am Ola. Now, please allow me to introduce to you our first speaker for today. Dr. Maria Jeneline Q. Diaz is a full professor and the current director of the Institute of Biological Sciences at the University of the Philippines, Las Bañas. She is also the R&D Program Director of the Philippine Genome Center, R&D Program for Agriculture, Livestock, Fisheries, and Forestry. Dr. Diaz teaches undergraduate and graduate courses in biology, genetics, and molecular biology. She has been mentoring students of BS Biology, MS and PhD students of genetics and molecular biology and biotechnology programs. Her research interests includes understanding the molecular mechanisms underlying the observed variation in coconut endosperms, development of molecular markers for various Philippine crops, and characterization of genetic diversity and population structure of various endemic native species in the Philippines. To discuss gene drive applications in food and agriculture, please welcome Dr. Maria Jeneline Q. Diaz.
Thank you, Janine, for that introduction, and a pleasant morning or evening or a pleasant day to all of you. And I would like to thank Isa for the invitation to speak in this webinar. So let me spend a few minutes in sharing my screen. For today, I would like to talk about gene drives, science and application on food and agriculture. This is the outline of my talk. I would first define gene drive and then discuss gene drive vis-a-vis genome editing. We would look into the mechanism of inheritance of gene drives, some types and uses of gene drives, and gene drives for pest control. I'm sure you have heard or read about gene drive, meiotic drive, driving Y chromosome, selfish gene, selfish genetic elements. So some of these terms have been interchangeably used for the past decades. And some of them, or they, if you actually look closely, a lot of them have overlapping definitions. Now, scientists have long known the occurrence of selfish genetic elements. So I think it has been decades since geneticists have discovered the presence of these selfish genetic elements. So essentially, I said that gene drives are naturally occurring systems and they are abundant in nature. And with the advances that's happening for the past decade or past number of years, and as Ma'am Ola mentioned, gene drive is one of those innovations that is picking up. So let us first define gene drive. A group of scientists actually proposed three definitions of gene drive. So this was in a publication in 2020 by Alfie and his colleague in PNAS. Gene drive can be defined as a process or a phenomenon. And as such, a gene drive is a process that promotes or favors the biased inheritance of certain genes from generation to generation. There would be prevalence of a genetic element or an allele of the gene, is increased. This leads to preferential increase of a genotype from one generation to the next and potentially spread throughout the population. So that's our first definition of gene drive. Gene drive can also be defined as a material object. As such, a gene drive is any genetic element able to bias its inheritance within a population. It is composed of one or more genetic elements that can cause the process of bias inheritance in its favor. But please take note that the presence of gene drive elements will not necessarily cause gene drive, necessarily cause gene drive. Some gene drives may confer preferential inheritance only when present in the population above a threshold frequency. Sorry, I'm having some difficulty. Okay, so let's now look into the third definition of gene drive. It is defined in the point of view of having an intention. So gene drive is a tool to effect certain changes in a population. It is intended to introduce new trait or traits into an interbreeding population so as to effect a change in the characteristics of the population. This may cause effects directly, for example, by inserting into and disrupting a target gene. Okay, so you see, gene drive can be defined in several ways, much similar to the different terms that we know. Again, it can be defined as a process, as an object, or as an intention. You must have heard, or if you have not yet heard of it, there is also the so-called synthetic gene drive. This is a gene drive system that is created through recombinant DNA techniques. So this time we're looking at this as a technology or a set of tools or a technique. But for synthetic gene drive to be done, two important criteria must be met or must be met in order to have efficient gene drive. The first one is organisms must have an inheritance pattern that can be biased. And typically it means that the organisms should be sexually reproducing. So with that, you already limit the organisms that can be modified through gene drive. For practicality, the organism must have a short generation time. Remember that gene drive's definition includes the spread of the gene drive from generation to generation. So it is quite logical to think that an organism with a relatively short generation time would therefore have a faster spread of the gene drive. But for organisms with a long generation time, that would be there would definitely be a longer period for the gene drive to spread. The use of gene drive has been there for so long, but then it picked up in for the past years because of the availability of the gene editing technologies like the CRISPR-Cas9 system. So the CRISPR-Cas system is actually viewed as a perfect tool to create a gene drive. And just to review, the CRISPR-Cas9 system includes three components. What you see in the left side, the upper left side of the panel, you can see there are the three components of the CRISPR-Cas system. You have the Cas9 endonuclease that is able to cleave or to make double-strand breaks in the DNA. You have the guide RNA, whose portions include a region that is complementary to the target and the guide RNA scaffold. And then you also have the PAM or the protospacer adjacent motif. This is found downstream of the cleavage site, which actually helps Cas9 to determine where the double strand breaks will be done. Now on the right side, upper right-hand side of the illustration, you can see there that once the Cas9 initiates the double strand break, the cell's own repair mechanism would kick in. So the repair mechanism could be non-homologous end joining method or homology-directed repair. In the case of gene drive, the homology-directed repair is the one that is used. On the lower left side of the illustration, you can see there a sample cassette of the Cas9. So you have there the Cas9 gene, the guide RNA, and then flanking Cas9 and guide RNA are homology arms. This cassette, when inserted into the genome, just like what you see in the lower right side of the panel, would mean that the Cas9 can now induce double-strand breaks, and after the repair system, you now have the homologous copy of the Cas9 and guide RNA together with the gene. Looking at it in another way, because at the moment, the creation of gene drives are now mostly based on gene editing. And this illustration is again showing you the target and the cassette, but together with the single guide RNA and the Cas9, you have the cargo. The cargo is a generic term that is used to refer to the desired genetic material that would be added to the system. And just like what I said earlier, once inserted into the genome, uh, uh, there will be double strand breaks, and then repair of the double strand break using homology directed repair. And in the end, you have the you have the transgene now, or the cargo together with the single guide RNA and Cas9 in a homozygous condition in the chromosome. So this is essentially how the CRISPR-Cas9 method is being used in order to create gene drives. So once the gene drive, or once the genetic element is in the genome, how would it be inherited? So let us look at the mechanism of inheritance. And the mechanism of inheritance of gene drives is best described if we compare it to the Mendelian inheritance. So on the left side of the illustration, you can see there the Mendelian inheritance, and on the right side, you can see there the gene drive. Let us say, for example, you have a mating between a heterozygous individual. So this is an insect mated. Let's say this is a homozygous recessive individual. So we know that Mendelian genetics tells us that a mating between a heterozygote and a homozygous individual would give you a 1:1 ratio in the progeny. And just like what you see here. Now, if these two progenies mate with homozygous recessive individuals or with homozygous individuals, you would still get that 1:1 ratio. So every time there is a mating between a heterozygote and a homozygote individual, you would get a 1:1 ratio in the progeny. But what if you have a gene drive? So you have a gene drive in this individual, which is heterozygote. In the first generation, you can see you have still the 1:1 ratio. But a mating between a heterozygote and a homozygous individual would give progenies that both possess the gene drive, as you see in this illustration. So you can see that as the generation goes along, you have increasing frequency of one of the genotypes, and ultimately it is possible that you lose the other genotypes, as you can see in this illustration. So for gene drives, the transmission to progeny is said to be at super Mendelian frequency, and that is greater than 50%. And if you look at the illustration on the graph here of the allele fraction against the generation, you can see an increase, a logarithmic increase of the allele as generations go along. So this is essentially the same illustration showing you Mendelian inheritance and gene drive inheritance. But what I would like to point out here is the difference in terms of the graph, the same graph that you had seen earlier. But you can see here the graph, when you have Mendelian inheritance, there is no spread. But with gene drive, you have spread of the allele and increase in the frequency of one of the genotypes. So let us now move on to some types and uses of gene drives. I actually chose only 4. I know there are some other types, that especially the ones that are being developed in conjunction or vis-a-vis with the CRISPR-Cas method. So the first one is the homing-based method. If I'm not mistaken, this is probably one of the older methods used for gene drive. With homing-based method, you have insertion of a gene drive nuclease with its own recognition site that cleaves DNA and then makes copies of chromosomes with the gene drive. So you can see there on the left side of the illustration, you have the nuclease. Okay, the gene for the nuclease, expression of that nuclease would mean cleavage of the DNA. And because of that cleavage, so again, you have your, you can have your double-stranded break, you have homology-directed repair, and then you would have a homozygous copy of the nuclease. The homing-based method is normally used to target essential genes. and for population suppression and population replacement. Later on, I would be talking or describing more about population suppression and population replacement. I just mentioned it here because when we look at the application of the gene drive system to pest control, you will meet population suppression and replacement. The second type is sex distorter or the Y drive, gene drive. This involves the insertion of gene drive to change balance between sexes. So it normally targets the X chromosome during male gametogenesis and therefore generates male bias. And in the process, it reduces the number of females. This is normally used for population suppression by changing balance of sexes. Then we have the split gene drive. The components of the gene drive this is called split because the components are split between chromosomes. And therefore, one spreads through drive, but the other through Mendelian inheritance. Therefore, it is generally used for limited target populations that are controlled in short periods and spatially limited or threshold-dependent control. The last type is the maternal effect dominant embryonic arrest or called MEDEA. This is a gene drive that involves a toxin expressed from a maternal promoter and zygotic antidote. So when the gene drive spreads, it causes the death of the organism that does not have the construct, since the toxin will kill any individual that does not contain the antidote. So sometimes you would also see toxin antidote system. This type of gene drive is normally used for population replacement. Okay, so let me now move on to, so after describing some types of gene drives or processes that you use in order to create gene drives, let us look at the application of gene drives in agriculture. If you look at gene drive, scientists thought that the best application in terms of in agriculture is really for pest control. So we know that pests such as weeds, rodents, and invertebrate species cause significant economic losses worldwide. So we are very familiar with this. We know that they are threats to agricultural productivity and food safety. So we know several diseases, several pests of various crops or even livestock, and we know how devastating the effects of these pests could be. So when we talk about pest control, there are two key population control methods. One is population suppression. This is when the spread of a genetic element that causes the number of individuals in a population is decreased, okay? Or you wanted to decrease or suppress the number of individuals, the pest individuals in the population. If you would recall the descriptions of the types and the uses of the types of gene drives, I mentioned suppression and replacement there, population suppression and replacement, because these are the two key population control methods. So you either want to suppress or replace the population. So again, in suppression, the spread of the genetic element causes the number of the individuals in the population to decrease. On the other hand, with population replacement, the spread of a genetic element through a population that causes a population genotype to change. So in some, they would describe this as population modification. So you replace, let's say, a genotype, or you modify the structure of the population, of the pest population. So with that, the use of gene drives for pest control has actually long been considered. So I think more than for several decades. However, it did not really pick up, not until the advent of the CRISPR-Cas9 technology. Because as I mentioned earlier, with the use of CRISPR-Cas9 technology, the creation of gene drives to control pests is more possible. And we know that the CRISPR-Cas technology is a very targeted approach. So you can really target the gene of interest and actually think of where in the genome you would like that gene inserted. One of the, or probably the very first agricultural pest in which gene drive was implemented is with the spotted wing Drosophila or Drosophila suzuki. So this was, I think, around 2018 or around mid 20s. So what I did was to look over the literatures and try to see where we are in terms of the use of gene drive with pest control. So if you look at the left side, this is a snapshot of a publication in 2023. So this is CRISPR-Cas9 based split homing gene drive targeting double sex for population suppression of the global food pest Drosophila suzuki. And then on the right side, I tried to look for gene drive papers on plants. So one is on insect. Actually, gene drive has been mostly used in insects. But in plants, I tried to look for some. This is one of those that I found out. So this is in Arabidopsis. We know that Arabidopsis is a model organism. And the title of the paper is Overriding Mendelian Inheritance in Arabidopsis with a CRISPR toxin antidote gene drive that impairs pollen germination. Okay, I would not really discuss the papers, but the reason why I'm showing this to you, so they are quite relatively recent ones, is to show to you that, and as I look into the literature, the state of the use of gene drive to control pests or the use of gene drive in agriculture at the moment is mostly on the development of methods, refinement of methods, to look at the utilization of this innovative technology in controlling pests. So you would notice that in both papers, it's they are actually describing methods or specific methods that would allow the suppression of a particular organism. So in the case of the Drosophila suzuki, the authors are saying that it is a cost-effective approach for suppression of the Suzuki population. And in terms of the Arabidopsis, this is actually, aside from Arabidopsis, I did not see any agricultural crops that are being, let's say, used right now to develop gene drive or to use the gene drive innovation. So with that, there really are opportunities. So gene drive as an innovative method really has the potential to provide significant benefits in terms of control of undesirable species. That's what I said earlier, that mostly people are thinking that gene drive can actually be used to control pests like insects. We know that insects, a lot of insects are vectors of diseases, and that also includes weeds. So if we do use, or if we are able to use successfully the gene drive technology to fight or to reduce pests, there would be the reduction of the reliance on pesticides and herbicides. Actually, that's one of the things that we really like. We want to remove or lessen our reliance on these pesticides and herbicides simply because we know that they are quite harmful to the environment. So with the reduction of the reliance on pesticides and herbicides, we can minimize environmental harm. A group is also saying that gene drives, particularly the endonuclease-based may be a valuable tool for working with polyploid species if it can be designed to drive edits across all homologous alleles and gene copies within a genome. So I think everyone knows that a polyploid organism has several genomes. And in a lot of instances, there are homologous alleles across the different genomes. If only we can design to drive edits across all homologous alleles and gene copies within a genome, then the gene drive method can actually be valuable for polyploid species. Even though there are opportunities, there are still several challenges. One, not all pest species are suitable for control using gene drives. Remember earlier I mentioned that gene drive can be used for sexually reproducing organisms. Okay. So not all pest species may be suitable for control using this methodology. Also, when we think about using gene drive, we have to look at the mode of reproduction. Again, it should be sexually reproducing, the mating system. So let's say, for example, for plants, a self-pollinated plants would have probably a slightly different methodology for cross-pollinated plants. We should also look at the fecundity, so the number of offsprings of every mating. Look at the ploidy level, because as I mentioned earlier, ploidy meaning you have several genomes. May prove to be quite, and it is actually difficult it is quite challenging to work on polyploid species. Also, genome size may affect the utility of gene drives. So when we design gene drive methods or experiments or systems in our research, then we have to take these things into consideration. Also, the identification of the gene targets. So what are the genes that will be targeted? That is important. So we have to know a lot, or we have to have a very good background regarding the species that we are working on, the characters that we are working on, what we need to target. And it will help a lot if you have the availability of the genome sequence of the organism. And we know that for crops, not all crops have available genome sequence, but of course the cereals, there are available sequences, but not for all. And also for plants, the transformation system for the organism is quite important. We all know that not all plants, or there are some plants that are recalcitrant to transformation. It's actually the same. It's the same with with the use of CRISPR-Cas method. You need to have a very good transformation system for the organism. If you don't have that good transformation system, then that would be a problem. Or the utility of the method would be quite a problem. More so, if you look at the challenges, the biosafety and ethical issues surrounding gene drive research and releases. So if you release if you release the gene drives, what would happen? What would be its implication on the evolution of the species, or the evolution of other species that it is normally in contact with? And also ethical issues. A lot of people would say that we don't want to make, let's say, a particular genotype extinct, or remove entirely a particular genotype. So again, there are biosafety and ethical issues that surround gene drive research and releases. Also, methods for controlling the spatial and temporal spread of gene drives and techniques to reverse their propagation in populations when necessary should be there. So we have to devise these methods that would allow us to control the spread, okay, the spatial and temporal spread of gene drives. Okay, and in some instances reverse the propagation in the populations when needed. So with all of this, uh, based on what I had seen in the literature, in terms of the application of gene drives in agriculture, we are still at the beginning. We are still at that stage that we are developing technologies, we are developing methods about the utilization of gene drive on particular species. I mentioned the case of the spotted white Drosophila, but I did not see any other example beside that. In plants, as I mentioned earlier, I have seen publications on on Arabidopsis, but none yet on crops, on important crops. So as I was saying, we are really at the beginning trying to look at this innovative method and trying to see whether this innovation can be used. It has a lot of promise. And we really want to take advantage of that potential. But then, aside from the challenges of the methodologies, there are also other challenges that I already mentioned. Okay, so I actually sort of simplified my presentation. So this is essentially the last slide. These are some of the references that I use, and I would like to thank you for listening.
Thank you very much, Dr. Diaz, for giving us an overview of the science and application of gene drive. Everyone, we look forward to your questions. You may send them through this Q&A Zoom function, and we will answer them during the Q&A session. Again, thank you very much, Dr. Diaz. Everyone, let us give her a virtual round of applause. Moving on to our next speaker, we have Professor Paul Thomas. He is a professor in the School of Biomedicine, University of Adelaide, and the scientific director of the Genome Editing Program and the SA Genome Editing Facility at the South Australian Health and Medical Research Institute. His research focuses on the development of CRISPR-Cas9 genome editing technology for a range of applications, including synthetic gene drives for invasive mammalian pest suppression. He has developed the first validated gene drive strategy for mammalian genetic biocontrol and has published over 130 scientific articles with more than 11,500 citations. To discuss the topic on fighting biodiversity loss with gene drive tools, please welcome Professor Paul Thomas.
Thanks very much, Janine. Thanks to you and Ola and Isa for the invitation to present on this very interesting topic today. And I'll just begin by sharing my screen. Can you see, Janine? Can you confirm if you can see that okay?
Yes, Paul.
Great, okay. And thanks also to Maria for that very good introduction to the topic. So what I would like to do today is to expand a little bit on some of those concepts that have already been covered, and give you some examples of our research and some research from others that have impacted in this sort of very new and emerging area of biotechnology. So first of all, invasive species, as we all know, are a massive problem. And a recent report just from 2023 indicated that the cost of invasive species globally each year is in the order of over $400 billion. And indeed, this cost has quadrupled every decade since 1970. So it's a big problem, and it's a problem that's getting bigger. In terms of invasive mammals, this is a significant proportion of invasive species. And just for example, in Australia, there's enormous damage to the environment and to agricultural productivity, particularly from mice, rats, rabbits, feral pigs, cats, and foxes. And in terms of the environment, many of the mammalian extinctions that have occurred in Australia in the last 200 years have stemmed from the impact of invasive mammals. So we need new technologies to address this ever increasing problem. And that's really the cause of the excitement around gene drives and their potential. So what I'm going to tell you about today, I'm going to cover a little bit of the territory that Maria has covered already, but then I'll get pretty quickly into some examples, particularly in the mammalian space. So I think we'll race pretty much through genetic biocontrol and gene drives, how they work. I'll just touch on CRISPR also. And then I'll tell you about some of the species where gene drives have been developed. We're talking really Anopheles mosquitoes and then some rodent work that we've done. And then I'll tell you about how we could apply gene drives to other mammals. And we're looking at that at the moment, really in the context of some fairly sophisticated modelling. And I'll show you some examples of that and some of the publications we have in that area as well. And then finish up with some challenges and barriers to gene drive development. So as I said, we all know about genetics and gene drive, especially now with that nice introduction from Maria. But just to quickly recap, if you look at the whole genome, and we're now looking at a mouse in this case, then there are 20 pairs of chromosomes that contain the entire genome, which is 2.4 billion bases. So 2.4 billion building blocks, making about 20,000 genes. And each gene provides an instruction to a cell. So a gene drive is, at least in those definitions that have already been presented, can be described as a genetic element. Often it's a synthetic genetic element that we introduce into the genome, for example, with this chromosome, and that gives the organism a new phenotype. In this case, just by example, it makes the organism glow green. But a gene drive is a special form of transgenic element that also biases inheritance. So what I mean by that is that, for example, if this is a gene drive now that's been integrated into one of the pairs of chromosome 1, it would mean that this chromosome has a biased inheritance, will be more often transmitted to the progeny than this one. And that's the basis of the whole gene drive modification or suppression strategy. Let me explain that in a bit more detail. And we've already covered this in the context of insects. It's the same concept in mammals. A gene drive will spread through a population because, unlike a normal gene, which only has 50% transmission, when it's a gene drive, it will be transmitted up to 100% of the progeny. And that's the extreme case shown here. And pretty quickly, you can see that the population can become modified. So that's a modification, which can be useful in some contexts. For example, if you're looking at mosquito control, and particular in the context of malaria, there's quite a bit of work going on to modify a mosquito population to see if it could become to carry a resistance protein, a resistance allele that would impact the population so that it couldn't carry the malarial parasite. So that's an example of a modification strategy. But most of the research that we're interested in, and indeed many others as well, is a suppression strategy. So how can we cause these gene drives to have a suppression strategy? Well, it's a bit trickier, but essentially what you do is you use the gene drive to spread through the population, and then you engineer it in such a way that once these gene drive carrying individuals meet, they can, for example, only generate infertile females. And this can cause the population to crash. Females particularly are, rather than males, are more, the females carry the reproductive load of a population. Therefore, most of these kinds of strategies will impact female fertility. And once you start to decrease the amount of females that are fertile, that will cause the population to crash. And there are various ways of doing this. The homing drive, as described by Maria, I'll touch on in a minute, but there are other strategies as well. For example, there's this so-called X shredder or driving Y strategy, and this is an example of that sex bias approach that was described earlier. So in this case, what you're trying to do is to make the population entirely male, and the gene drive only generates male progeny. Therefore, you get an increase in males over females. This will also cause a population to crash. And these are ways of suppressing a population using a gene drive. So the enormous potential around this technology really is several-fold. Well, one is that it's a non-lethal approach. So with increasing concern around the use of baits, for example, if we're talking about rodents, which, is a rather toxic control method that causes pretty, nasty form of death. There's ever increasing interest around, using alternatives to this, to the traditional bait and toxins. Secondarily, gene drives are a landscape scale approach, which means that we could affect an entire population with relatively few introduced individuals, and which is, likely to be a cost-effective way of suppressing a population or modifying it. And lastly, because it works through genetics and breeding, it's species specific, as only individuals with the same species can generate fertile offspring. So why do we want to introduce gene drives? Well, I guess the context of this more broadly, beyond the environment and agriculture, also extends into human health. And a lot of the drivers around this work are to generate new methods for malaria control, as it's such a huge burden in terms of human health impact. There we go, laser pointer. Yeah, so around 500,000 deaths per year are caused by malaria, which is huge. In the environmental space, we're interested in rodent control, and that's because rodents on islands particularly cause devastating impacts on bird populations in particular. And also in Australia, many of you might have seen the videos of the plagues which occur every 4 or 5 years or so. So Australia has a lot of grain crops, and when conditions are right, mice breed up in enormous numbers and essentially completely take over those grain cropping regions, and literally millions of mice are cause devastation in those areas, which not only is, of course, a huge impact on the crops, but also the people that live in those regions. So CRISPR-Cas9, I won't go through this again, except that it provides a tool not only to make the animals that carry the gene drive, but also for the gene drive itself to work. And you can think of this complex really as a pair of molecular scissors that enable us to cut virtually anywhere within the entire genome. So a very powerful technology. And as Maria mentioned, it has been a deal breaker in terms of translating some of the ideas around gene drive research, which have been in the literature for decades, now into sharp focus, so that we can actually start to test some of these concepts out in at least model organisms. So there are various strategies, and again, I'm not going to go over these in detail because we have heard about them previously. Most people, when they hear of gene drives, are probably thinking about this so-called homing drive, which is the one that's working pretty nicely in insects. So this is a way of modifying the germ cells so that they'll become homozygous for a gene drive by causing a double-stranded break and then copying over of the gene drive element, but just in the germline cells, which means that it always transmits to the next generation. So to cut a long story short, this mechanism, so it's called a homing mechanism for historical reasons, but this, for whatever reason, works particularly well in Anopheles mosquitoes, which is fortunate if you're working in malaria, and it's great. But other species don't tend to have this working as efficiently, and that includes mice. Rats, there's some evidence that the homing mechanism is much better than mice, but really, probably even for mice and rats, it's not at a level which would enable us to modify a population with a great degree of efficiency. So people are still working on this, but suffice it to say that we and others have looked at homing drives in mice, and we've pretty much just put it on the shelf for the moment, and we'll come back to it if it looks more promising in the future. The second approach, which again you've heard briefly from Maria, and has been developed to some degree in insects, is this so-called driving Y or X shredder approach. And the concept here is to ultimately to modify the Y chromosome so that it contains this gene drive element. And in this case, the gene drive element goes to work during sperm development, and it will target the X chromosome during sperm development whilst the X and Y developing sperm are still connected, and target it in such a way that the X chromosome becomes inactivated through cleavage by the CRISPR-Cas9 complex, which ultimately means that the X-bearing sperm are inactivated, whereas the Y-bearing sperm, which contain the element, remain active. And this means that you get transmission of the transgenic element, the gene drive, to all of the progeny, and also they're always male because they contain the Y chromosome. So it's a lovely concept, and it has, as I said, been working reasonably well in insects, although there's still challenges that remain. This is really just to bring you up to date in terms of mammals. Of course, mammals also carry an X and a Y, and you could think about doing this kind of approach in mice, for example. And we had a very decent crack at it. We spent quite a bit of time and effort to try and develop this approach. To cut a long story short, we could engineer the mice so that they had all of the components that they needed to have this occur. But when we activated the CRISPR-Cas9 complex, and it did indeed cleave the X chromosome, instead of biasing the sperm offspring, at least in mammals and at least in our system, what happened was that we inactivated the spermatogenesis program completely. So here's a wild type mouse testes, and what you're looking at are essentially little sperm factories. These are tubules, of which there are thousands within the testes, and there's an outside to inside differentiation program whereby the sperm that are mature end up on the inside in the lumen of these sperm, and then they're eventually released into the ejaculate. Here's an example of one of our X-shredder mice. This is the more extreme example where we've really cleaved the X chromosome many times. You can see that these tubules, which are still present, are essentially empty. So there's no active spermatogenesis because of this cleavage event that we've engineered during spermatogenesis. So again, this strategy we think is probably one that won't translate from insects into mice. And again, we've shelved this shelved this for the moment and are pursuing other avenues. So this last avenue that I want to tell you about is really the most successful approach that we've had to date. It's the first example of a mammalian gene drive strategy for suppression, and it's based on a natural gene drive called the T haplotype, which we've engineered to generate so-called T CRISPR mice that target female fertility. So how does it work? So as I said, somewhat I guess, as an alternative to using strategies that have been developed in insects, we targeted this strategy because we knew that there was a natural drive element already in mice. So this is a mouse-specific drive. And the natural element is really a variation of just the normal chromosome 17. So this is in schematic form here. Here's just the normal chromosome 17 in mice. It's around 100 megabases long, and this region shown here is the region that's of interest to us because of a natural variant called the T haplotype. So the T haplotype, it just refers to really about a 40 megabase region of chromosome 17 that has evolved naturally to have a very high inheritance bias. So what I mean by that is a male mouse that carries, as you know, there are 2 copies of every chromosome. So a male mouse that carries the normal chromosome 17 and the T haplotype version of chromosome 17 will pass on this T haplotype up to 95% of its progeny. whether they be male or female. So a very high natural inheritance bias. And we know to some degree how this works, so the molecular details of it, although that's an area that's gaining renewed interest now, and including us, where many groups are looking at it. I don't have time to get into that today, but the important thing here is that it's a natural gene drive element that we can use and manipulate to see if we could, to turn it into a suppression tool. So that was essentially the question: can we manipulate the natural T haplotype in such a way that we can use it to suppress invasive mice? That's the aim. So I'm going to describe a lot of mouse engineering really in a very simple way, and it doesn't quite do justice to the amount of time effort that was put into this, but it really just describes the concept. Okay, so in my laboratory, we have the ability to engineer our own mouse models. So we imported the T haplotype mice from a collaborator in the US, David Threadgill, and we engineered it by inserting into the T haplotype a CRISPR cassette. So this is a gene expression cassette that encodes the CRISPR machinery that will target a female fertility gene. I'll show you how that works in a moment. But the important thing, first of all, was that we've now manipulated the T haplotype by inserting a new genetic element into it. And we needed to confirm that that didn't alter the inheritance of the T haplotype itself, because we still wanted its natural drive. And indeed, we showed that even though we'd added a little bit of extra DNA, this was completely benign, and the T haplotype maintained its normal biased inheritance, which was great. That's what we were hoping. So that was good. So what does this CRISPR cassette do? Well, it targets a gene on a separate chromosome, a completely different chromosome, but this is a well-characterized gene that is required for female fertility. And it's a so-called haplosufficient gene, which means that you need to knock out both copies in females for it to cause female-specific infertility. And the concept here is therefore that in males, all the action occurs during spermatogenesis. We activate the CRISPR-Cas9 and knock out the female fertility gene. This doesn't impact the males at all because it's a female fertility gene. So we get high bias transmission of the modified T haplotype, which we call T-CRISPR, and at the same time, we get high transmission of inactivated version of the female fertility gene. And we chose prolactin as our example gene, but it could be any haplosufficient female fertility gene. And when we looked at the progeny, they were inheriting this transmission of inactivated gene at about 80% frequency. So that's what we'd managed to achieve in the lab. And we thought that was pretty good, but we didn't know whether it was going to be enough to cause suppression or not. So we then turned to a pretty sophisticated model to figure that out. So what do I mean by that? So up till now, I've just been describing the experiments that we're performing in the lab. So now I'm going to describe to you a model that we've developed to try and get the best understanding about how this engineered material from the lab would behave in an environmental situation in the real world. And of course, it's only a model, but nevertheless, we try and make it as realistic as possible. So essentially, without going into too much detail, I'm not a modeler. It's not my background, but essentially what we do is that we keep track of around 200,000 individuals in a computer simulation, and we keep track of them in terms of their genotype, their mating profile, their sex, and all of the other properties which we can get from the field, which would dictate how they would behave in the wild. So they go through a cycle, which is their normal life cycle, a mate search period. They have offspring. There's dispersal. There's a survival element because some of them won't make it through in the wild to survive to the next generation. And then they go through the whole period again. So it's basically this cyclical model where we track around 200,000 mice in a defined environmental space. So it's a spatially explicit, individual-based stochastic model for those of you who are more familiar with this area. And this is work by Ashigul Barand, who's really led this part of the research. So I'm going to show you some data because I really like these videos, and they sort of really give you a better sense of I guess the potential of the technology. Oops. So what we're going to look at here is a hypothetical island. We've got 200,000 individuals. Each dot represents a patch of mice, so it's like a little local population. But as you can see, they can disperse between patches, and we're going to put into that 256 T CRISPR mice, and this is based on the data we've generated in the lab. So we're trying to simulate what would happen if we put these lab-based, engineered mice. If we could translate that to the environment, what would it look like as a proof of concept? And the gene-drive mice, as you'll see in a minute, are, oops, I don't know why that keeps going forward. Let's see if we can make it start. The gene-drive mice are, I'll go from the other way. Oh, here we go. Okay, good. So the gene-drive mice are in red. So, okay, we've just popped the gene-drive mice into the population, and critically, they're building up pretty quickly. So we're now increasing the number of gene-drive mice, so that's T CRISPR mice, and then you'll see the size of the red dots will decrease, and that indicates that the population is getting smaller and smaller. So over a period of time, there's initially a spread, and then there's a contraction of the population, and eventually, as you can see, we've completely eradicated. And that, when we see that, that's a very encouraging sign for us in terms of types of drugs we're trying to develop. If you want more detail about this, I'm happy to answer questions, of course. The paper has now been out for a couple of years, and to give you an update of where we're going next, this is the first proof of concept for a mammalian gene drive. We're now generating an island-specific version of it. So, as Maria mentioned, one of the big challenges in this area is to target the gene drive in time and space. We don't want to affect populations that are not the target population. And one way that we've thought of to do that is to exploit a genetic predisposition. For example, an island fixed allele. So in other words, a gene on an island, an allele that's got a particular sequence to it that's not homozygous or not fixed in the mainland population, which means that we can go after the island mice in a quite a specific way and not worry about having to impact the mainland mice, even if there's some escapees from the island. Happy to answer questions on that if there are any. There's a lot of work going on with stakeholder and regulator engagement, and also we are part of the GBIRD International Consortium, which we have lots of really interesting discussions around gene drive tech for all sorts of applications. So lastly, what about other invasive pests? I've focused on mice because of the obvious reasons around, especially in Australia with the plagues, but what about others? So and as mentioned at the start of the talk, many of these mammals have a really devastating impact on the environment. Gene drives have been discussed a lot in the literature, and many see them as a really technology that's full of potential. But in reality, can we get a better sense of what it would take? How long would it take if we could engineer these animals for them to have an effect in the environment? So we did a pretty close look at that in a recent paper. Again, this is work from Ashigul. And to cut a long story short, we looked at some of these classic drive mechanisms, which, to be clear, have not been engineered in these animals yet. But we did this in a hypothetical way to ask the question, if we could engineer them, how potent would they be? And we kept the population size in each case the same, which is the way that we felt was the best way to do it, at least to get a comparison data set. But what that means, interestingly, is that because of the density of these larger mammals being much less dense than rodents, it means that we're talking very large areas when we're talking about a population of 200,000. For example, the cat simulation, which I'll show you in a minute, is in an area around 100,000 square kilometres. So that's huge compared to, say, the 20,000 square kilometre island that's the same that is the size of the 200,000 population that we looked at in mice. So again, without going into too many details, and the publication is out, so you can look at it. We've tried to, as best we can from the literature, ascertain all of these key parameters to make this as realistic as possible for these larger mammal species. And this is what we got. So this is an example of 256 gene-drive cats being introduced into a population of 200,000. And as I said, this is a huge island. For those of you who know Tasmania in Australia, it's about the size of Tasmania and a half, so pretty big. And the question was, does it work? And as you'll see, it can work, but it takes much longer than it does in the smaller animal populations, as you would expect. And as Maria alluded to, the longer generation times of the larger animals and often the smaller litter size means that we're looking at longer eradication times for an equivalent population in terms of number. And in this case, we're talking, many, many years. We're talking over 100 years, probably closer to 200, although this is a bit of an extreme example. If we directly compare, it's probably at least 5 times as long to eradicate the same number of larger animals as it is compared to, for example, rodents, mice, and rats. The interesting middle ground here is rabbits, and rabbits are a big problem in Australia. Again, they've run rampant since in the last 200 years, and since they were introduced. And in terms of modelling, they're I think a pretty reasonable target to go after, and probably the animal that I would pick after mice. The next would be rats, and I would then go for rabbits. And we're having some discussions about that at the moment. Okay, so to conclude and just give you some of the final, updates and challenges, I think it's fair to say that genetic biocontrol, gene drive technology is progressing really with a lot of interest in many labs in the insect space as well as now in the rodent space because of its huge potential for disease control, conservation, and agriculture. There are many, many facets to this research beyond which I've had a chance to tell you about today. There's a lot of stakeholder engagement that is and will need to be done in order to, to make government and the public aware of its emergence and its use, and some of its, the caveats around its use as well. There's a lot of regulation going on as it's very new and highly and a form of GMO that can spread in the environment. So that is certainly an area that needs to be regulated. There are lots of technical hurdles that need to be overcome as well, including this ability to target specific populations. And as I said, we're looking at ways of doing that at the moment. In terms of larger mammals, we're looking at longer time frames than rodents. There are technical challenges that really stem from the fact that many of these larger mammals are not model species, so we don't really know how their genetics works as well as some of the model species like mice. We don't know their reproductive biology as well. There are less facilities to house them, and the capacity to make transgenic organisms, again, as alluded to by Maria, is less well defined. And in some cases, for example, cats, they're also domestic non-model animals. So that's another element of, societal acceptance that would have to be overcome. So with all of these challenges, though, I think I remain very positive about gene drive technology in the animal world. And the last comment I want to make is just around this fairly new pair of papers. So Maria mentioned, I think the one on the right-hand side, there's a companion paper in the same issue, I think, Nature Plants from Bruce Hay. And essentially these are two forms of a gene drive. They're called cleave and rescue drives. Again, I'm happy to discuss these in more detail. Really interesting approach, and notably the first examples of engineered plant gene drives that I'm aware of anyway. And the transmission bias is quite remarkable at over 90%. How they're going to translate into other plants, for example, weeds, is yet to be established, but nevertheless, the technology has taken root, if you will, are now in plants as well. So with that, I'll just finish off by thanking my team. I spoke about a lot of work. The mouse work particularly is done by a fine PhD student who's just completed, Luke Giras here on the right-hand side. The other gene drive members of the lab are Gelshan Godahua, as well as Louise Robertson, and Sandy, who is here, is our chief mouse engineer. She does all the injections to make the transgenic mice. We've had lots of very generous sponsors and collaborators as well. And with that, I'll close, and I'm, I guess, with Maria, happy to take any questions. Thank you.
Thank you very much, Professor Thomas, for that very informative presentation. Definitely, the presentation of our speakers has given us more knowledge about gene drives and its applications. Again, thank you very much, Professor Thomas. Everyone, let us give him a virtual round of applause. So that concludes our presentations for today. We encourage everyone to post your questions through the Zoom Q&A. For our Q&A session, we have Mr. Panfilo de Guzman, Senior Associate Scientist at ISA Inc., to moderate the discussion. Sir Popen, the floor is now yours. Sir Popen, I think you're still on mute.
Thank you very much. Okay, thank you very much, Janine. We have a lot of interesting questions posted on the Q&A box. And this one is addressed to both speakers. The question is, there might be an issue on ecological balance. It's either for extinction or on the extreme side. If we overkill an organism, how do you address the issue? And I think question related to that is there a way to control certain gene drives so as not to entirely wipe a population? Professor Thomas, please, and then we can Dr. Diaz can add on that one.
Great, thanks, Vivek. It's very interesting questions. The second one around spatial control of gene drives, and so that it doesn't affect the entire population, and a timing mechanism, if you like, within a population, is one that hasn't really been solved yet, even in organisms that are in which gene drives have been more extensively developed. For example, Anopheles mosquitoes. There are forms of gene drives that Maria alluded to that are threshold drives. So this is potentially a way, particularly in the insect world, where, if I were to back up for a minute, there's gene drives, as we've been describing them, sort of at one end of the spectrum. And at the other end of the spectrum, there's technologies like sterile insect technology, where you really swamp the population with sterile males. And there's no form of gene drive, no form of spread of a genetic element, but rather you're just using sheer numbers of sterile males to limit the amount of females, because once they breed once, they then drop out of the population. So in between those two extremes, there are threshold drives where you can introduce, particularly in insects, a sufficiently high number of individuals so that they're above a certain threshold in the population, and then the drive will become self-sustaining. So they're the kinds of approaches that are probably going to be the most amenable in the insect world to have limit in time and space, because you've got that threshold effect. So if a few individuals get off into another population, there'll only be a few, therefore they won't be able to sustain the drive, they'll drop out. That's quite an attractive idea for insects. It's not really feasible for the mammalian systems that we're working on. I could briefly answer the first part of the question. If you could just remind me quickly, what was that? What was the concept there? It was about.
Let me. It's about extinction of the population.
Oh yes, yeah. I just wanted to say with that one, we're mostly focusing on invasive species. Therefore, there are species that it shouldn't be in the target areas that we're going after. So I think we know about, to some extent, about what that population would look like without those invasive species, and at least in the context of islands, traditional eradication using baits of rodents on islands produces huge benefits, and that's kind of not controversial at all. So we kind of know what the effect of removing the other rodents from the islands would be, and it's good. And I think what's less clear, though, is perhaps in, for example, malaria, when you're targeting a species that's normally within that environment, the mosquito, what happens when you take that, although it is carrying malaria, but if you take that species out of its natural environment, it's a bit less certain as to what the outcome of that would be. And I think that's something that target malaria, for example, are looking at pretty closely. Thanks. Over to you, Maria.
Yeah, thank you very much, Paul. I think Paul was able to answer very well the second question, so I don't have any addition for the second question. For the first question, I do agree that it's good for invasive species because, as Paul mentioned, they should not be there. So that is actually I mean it's sort of it validates or it's fine to sort of eradicate that invasive species in the region. But for those that are natural parts of the ecosystem, that would be something that has to be looked at, I think. Because we don't normally, or sometimes we don't normally know what would happen if you really remove one species in a particular ecosystem. So I think that's one of the things that if somebody would release, let's say, a gene drive system into the environment, that's one of the risk assessments that should be done.
Thank you. There's a question now also addressed to both of you. Can we say that most gene drives are sex-linked? I think the question is referring to that it can be applied only to sexually reproducing species. Is this really a humane tool for population suppression? So that's a question. Dr. Garcia.
Yeah, I can just probably briefly say something. It is really, I mean, more applicable for sexually transmitted, sexually reproducing species, because as we know, there should be transmission of the trait, and that happens when you have the sexual reproduction. So, but I think not all gene drives are sex-linked. I think that's one of the questions. Not all gene drives are sex.
Professor Thomas,
yes.
Do you have anything to add?
Yes. Yeah, I agree. Actually, one thing that was of interest on this topic that came up in a workshop that we did a few years ago that we hadn't really thought about was that the driving Y or X shredder approach, as we both described, biases the population towards males, and therefore you get a crash because there's not enough females to sustain the population. We didn't really think about that balance of the population where there'd be an excess of males and therefore it's great competition for the remaining females as the population declines, and how that could affect the behavior of the animals. And that was actually one of the key concerns that came up from the stakeholder group, that perhaps although we sort of say this is a very humane approach, which it is in many ways because it doesn't use a toxin, on the other hand, we need to be aware of changes of behavior and pressure on mating from the limited number of females that are available, and how that might affect mouse behavior. So that, and potentially other species. So yes, it's a fair comment, and one that I think should be part of any risk assessment and will be certainly looked at pretty carefully if that kind of drive is one that goes forward.
I have a follow-up question here, maybe related to one. Can the gene drives be used to alter the genetic makeup of bacteria and viruses? Do you have any idea if there is some research on that? the application of gene drives on bacteria and viruses?
Yeah. So, bacteria already have systems that enable sharing of DNA between bacteria. So it's kind of not apples. It's a different world. There's a single bacterial chromosome. It's not there in a pair of chromosomes. It's much smaller. They can share DNA via plasmid transduction already. So it's kind of the way the system is set up is quite different. So that these gene drive concepts that we've been talking about are really, I think, eukaryotic specific. I would say, the way we presented them doesn't mean that you can't get something similar happening in bacteria, but not the way that we've quite described it. Maria, do you want to comment on that?
Yeah, there might be a parallel system, but not exactly, as Paul mentioned, not exactly the way we discussed this.
Mm-hmm. Okay. So maybe a related question also. Would engineered gene drives work on any species, or could gene drive affect human populations, and what are some of the potential challenges of developing gene drives? Can it work on any species? Can we apply the gene drive tools to any species?
I think I mentioned earlier that it is mostly applicable for sexually reproducing species. In case of humans, I think we would have a lot of ethical issues on that. So, I think we have to look into that first.
Professor Thomas, do you have anything to add?
Not really. I mean, you can't capture gene drive from the environment. So, in other words, it's not.
Yeah.
You would need to engineer a species for a gene drive to work. So the only way it could work in humans would be to engineer a human embryo, which is illegal, and therefore I can't really see this as being a problem we're likely to face.
Okay, thank you very much. There's a question here for Dr. Garcia. What are the expectations on the difference of gene drives applications in polyploids and diploids? And is there any visibility gaps between the two, between the polyploids and diploids?
I think I was the one who mentioned the use of gene drives for polyploids. So at the moment, the gene drives or gene drive technologies they are being used for diploids, and it would be a bit more challenging to use it for polyploids. In fact, as Paul mentioned, the gene drive in plants has just started. So, and a lot of plants are actually polyploids. So I think development of the technology that would allow the tinkering of the polyploid genome with gene drive would still have, they have to be developed first. Definitely, it's a lot more challenging to work on polyploids compared to diploids.
There's another question here about can the technology be used to protect species at risk of extinction, and what factors would need to be carefully considered before using the technology? The question is to both of you. Can we have the answer from you first, Professor Thomas?
Yeah, it's an interesting one. It's been so. Can we use gene drives to protect species that are close to extinction? Personally, I'm not sure that is practical or ever likely to be an option. And I say that because most species that are close to extinction are not species that we can easily and naturally manipulate anyway, and let alone develop a gene drive, which we're talking, many years of development before it's ready. So to me, the idea of taking a threatened species, establishing a colony, engineering it, proving gene drive efficacy and then releasing it in such a way that would somehow protect it against the predator or the environmental influence is really not likely to occur. I think we're much better off trying to develop gene drives for the invasive species that are doing the damage and letting the threatened species recover from the lack of those invasive species rather than the other way around.
Yeah, I don't have any addition. I agree with what Professor Thomas mentioned.
Mm-hmm. A follow-up question, perhaps. Is there a possibility that gene editing approach can, in controlling pest population, will result in to mutation for the gene-derived species? Professor Thomas, please.
Yeah, okay. If I understand the question correctly, it's a good one. So because there's a few aspects to it. Give me a minute or two. So because we're using the CRISPR machinery to induce gene drive replication to enable it to spread, it doesn't always work as intended. We know that from lots of experiments. And sometimes instead of getting, for example, copying of the element over onto the other chromosome, it just makes a small hole in the chromosome, and that's a mutation. And that is a so-called resistance mutation from then on, because it's not susceptible to the CRISPR machinery anymore because the sequence has changed. So those resistance mutations actually stop gene drive spread. And there's lots of work going into ways of getting around that, and you can get around it, but it's something that we do need to consider carefully. So that's one thing. A second thing is that sometimes naturally within a population there's polymorphism, so in other words, variations of the genome that are already present that will just stop the gene drive from working because they've got a slight sequence tweak or alteration at the target site. So if they're already in the population, then that's something we need to be aware of so that we can counter their presence, because otherwise they'll prevent the gene drive from spreading. So they're natural resistance mutations. The third point quickly is that CRISPR, although it's very good at cutting where we hope it's going to cut, sometimes it will make a mutation in the genome that's not the intended site. That's called an off-target cut. And those off-target cuts could potentially decrease the fitness of the organism that carries the gene drive. So that's another aspect of testing that has to be, sort of established as part of the gene drive development. So there's sort of three examples of where changes in DNA, either natural or CRISPR-based, that can affect gene drive spread.
Okay, thank you very much. With that, is hybridization with a related gene drive organisms possible? Dr. Thomas? Prof. Thomas, please.
Well,
do you have an answer for that? It's a very technical question, I think.
Yeah, no, it's a good question too. So this is the question about whether a gene drive could spread to another organism. So I've always thought about this as being a situation where if you've got two, only individuals from the same species can generate fertile offspring. Therefore, a gene drive will stay within the same species. That's my simple answer to the question, and I'm happy to get pushback on that or an alternative view. I'd be interested to explore it, but that's always been my view on it. Maria, do you have anything to add?
No, I don't have any answer. So
here's a very interesting question about public acceptance. How do we effectively address and manage negative consumer reactions to gene drive technologies? Professor Thomas, do you have any experience about public perception about the technology?
We collaborate with real social scientists that are exploring this area, and it's a crucial thing, because I don't think we'll ever get to the point of deploying anything, even if it's the best gene drive in the world. If the public don't accept it, then the government won't accept it. It won't be used. So how to do it? Well, I sort of favour an open model. Like we try and develop the technology in a transparent way, in the hope that brings confidence in the process. I think people are justifiably wary around GM, in the sense that in the past it hasn't always been simple, and there have been the impact of industrial pressures on GM have and commercial outcomes have meant that it doesn't always have a good name. So I think we need to be very conscious of that. So I'm all for transparency. I'm all for engaging early with the public, keeping government informed. We are quite strictly regulated in Australia in this tech, and I think sort of all around the world that would be true. So all of those things together, I hope will bring confidence that we're not a bunch of mad scientists trying to cook up a sort of a technology behind closed doors for a commercial gain, but rather we're in it for the public good and the environment's good, and trying to really bring a new technology to bear on a pretty terrible problem of invasive species. So that's the way I hope about it. I know there will always be elements though that find the technology concerning. I think the best thing to do is to engage early and genuinely, I suppose, with those groups.
Do you have anything to add, Dr. Diaz? What is the current situation in the Philippines? Are there any issues on acceptance about the technology?
Yeah, personally, I think what is happening right now with GM, with no plant breeding technologies in the Philippines, likely we would have the same problem with if ever we decide to have a gene drive, or to have to introduce gene drive in the Philippines. So I think it would just be, it would be the same, and I think we can just do what we are doing at the moment, engage everybody. The government should be well informed, as mentioned by Professor Thomas. So I think everything that we are doing at the moment, and if we can add more, then that would hopefully make it more acceptable.
Okay, thank you. This question is particularly addressed to you. How could the implementation of gene drive technology in the Philippine agriculture impact the traditionally older, less technology savvy farming community, and what challenges might arise in adopting such advanced technology?
Yeah.
Dr. Garcia.
Yeah. So I think if in case we would have gene drive in the Philippines, the risk assessment would be it would be a very important part of this, especially when it comes to the effect on the environment, to the effect on the natural ecosystem, the existing species in the region. So likely it would be like what we have or what we are doing with GM crops, and that's what I can think of at the moment. So really a lot of risk assessment is needed for this.
Okay, thank you. And a question to both of you. Do you think gene drives can be used to improve polyploid crops, to improve as an economic for economic purposes? I mean, maybe improving the traits of the crop using gene drives.
I think I mentioned earlier that it would be challenging, but who knows? In the future, there might be methods that would become available for using gene drive in polyploids. Let's look at sequencing. Sequencing has been so difficult with polyploid crops before, but nowadays that is not much of a problem. So I would not be surprised, let's say, that in the future we would have technologies that are developed in order to address that problem.
I think this one,
Oh.
this question is, do you have anything to add,
No,
Professor?
I was going to say no
Okay.
more to add.
Okay. This is the question is it wise to put CRISPR in the genome of mosquitoes to suppress their population? Is it possible that mosquito will change the payload payload to survive instead of suppression? Anyone can answer the question, please? Dr. Diaz?
Okay, that's an interesting question. So if I get it right, instead of being suppressed, the mosquito would sort of adjust or adapt to the presence of the gene drive. Did I get it right? I hope I get it right. So, okay, I'm not sure how to think about this, but with gene drive, there are really specific mechanisms that would allow the super Mendelian transmission. So as of now, I think that would be a different. I mean, at the moment, I cannot think of any mechanism that would allow I mean, instead of having suppression, you have sort of an adaptation to the payload. So, but so I'm not actually sure.
Yeah, it's hard to imagine how that would work. The suppression strategies mostly act by having the gene drive inserted in a female haplosufficient fertility gene. So in other words, there doesn't even need to be a cargo beyond the CRISPR-Cas9 machinery for the suppression strategy to work. So, yeah, so that I think means that unless the gene drive changes its location somehow, then in which case it won't have a suppression effect anyway. So I can't see a way of it. I can see a modification strategy being affected somehow by the cargo, but not a suppression strategy. But anyway, it's all a bit hypothetical, I think. The other thing I would say is that the cage trials that have been done with pretty large numbers now have managed to see eradication with thousands of mosquitoes. So at least in that population size, there's nothing at least reported that's sort of indicated that there's huge instability of the gene drive element. Of course, it doesn't mean we don't know what would happen with a larger population. But anyway, it will be interesting to see whether in very large populations we get some quite strange adaptations to gene drive, and its molecular configuration that are unanticipated. So I think it's a good thing to be thinking about for sure.
Okay, thank you. Could gene drive work with sexually reproducing species where sex is determined by environmental factors such as temperature? Professor Thomas, do you have a answer to that one?
Yeah. Yes, it could, although we haven't modeled it and haven't seen any modeling on that. So I guess you're thinking about some reptiles where, temperature of the egg, for example, can affect the sex of the offspring. I haven't seen. Yes, in theory, but I can't give you any details or point to any studies that have looked at it. That's about as much as I can say.
Dr. Garcia, do you have anything to add, or you have any? You don't have any additional info on that one. Okay. Can we use gene drive to favor population shift from one host plant, example crop, to a different host plant, say a wheat? That's a question from our anonymous attendee. Can we use gene drive to favor population shift from one host plant to a different host plant, from a crop to a wheat, for example? That's the example that was given. Anybody can answer, or?
I'm not sure whether I also got this correct. Does it mean that instead of, let's say, the pest infecting one host plant, it will shift its infection or its infestation to another host
Very
plant?
correct. Yeah.
Okay, yeah. So gene with gene drive, so you would be looking at genotypes. So what I can think of at the moment is that if there is a specific genotype that would be infecting or infesting a particular plant, so maybe a shift in the genotype can be done. I mean, you favor one genotype against the other. If you can find a gene and the alleles that would make that possible.
Oh,
Sir
you're on.
Popen, you're on
Unmute.
mute.
Oh,
You're
sorry
mute.
about that. Yeah, okay. Another question here. Which application of gene drive is currently in advance and would be more acceptable to health, food and agriculture, and in controlling invasive species? So, which gene drive tools are more advanced when it comes to health, application in health, food and agriculture, and in controlling invasive species? Can we hear from Professor Thomas, please? And then
Yeah. I would say undoubtedly the health sphere because of the malaria and so Anopheles based malaria control as well as Aedes mosquitoes based dengue and other viruses. That's been a big driver, but it's mainly malaria, mainly. And part of that is this very fortunate high efficiency homing that they see in Anopheles mosquitoes, which has driven a lot of that not only urgent sort of human health need, but also has made it technically possible to test many different sort of configurations of gene drives. And there's quite a lot of advanced versions of that homing drive, particularly that have been conceptualized and then tested, and that's going pretty well in Anopheles mosquito. So that's where I think the major advances have been. Of course, there's like this Drosophila suzuki experiments that were mentioned by Maria, and our work in rodents, but really a lot of the main activity has been driven around malaria.
Okay, thank you. This one has already been answered by Dr. Garcia. But for the interest of other participants, the question is it ethical to reduce into extinction or control an organism just because it is considered a pest? How about the possible gene flow and loss of biodiversity once it is commercialized? Can you answer that online, live, Dr. Garcia, please?
Okay, so again, so whether I'm sorry, I did not get the question. So
On the ethical issues, is it ethical? Yeah, is it ethical to reduce into extinction or control a particular species or organism?
Yeah, I think we sort of answered this a while ago also.
Yeah.
When it comes to invasive species, it is, since invasive species are supposedly not on that location, so they are not native to that region. So removing them or let them because anyway it's just in that region. Okay, it's not all over. So for that region, maybe you can have it removed. But I think we also mentioned earlier that, but for naturally occurring organisms or systems in a particular environment, it would be quite difficult to do that. So and as I said, I think earlier too, that we have to do some risk assessment regarding the effects of making a particular species extinct in a particular region.
Okay, thank you very much. There are still a lot of interesting questions, but because of time constraint, we will move to our closing. I'll give back the virtual. Thank you very much, our resource speakers. Maybe we can have a virtual round of applause to them for answering the questions. Okay. Thank you, Professor Thomas and Dr. Garcia. I now give back the virtual microphone to Janine. Janine, please.
Thank you very much, Sir Poppen, for moderating the Q&A session. We encourage everyone to please complete the evaluation form to receive your certificate of participation. Scan the QR code flashed on your screens or go to the link to access the evaluation form. This post-webinar survey will help us improve our future events and webinars. Again, to receive your certificates, you need to fill out the evaluation form. For everyone's information, we will be having the third part of this webinar series in January next year, focusing on novel foods. If you are interested in joining the said webinar, please do follow us on our socials at isa.org or subscribe to ISA's weekly newsletter, Biotech Updates. Stay tuned for our future announcements, and we hope to see you there. Before we wrap up our today's webinar, we extend our sincere gratitude to our resource speakers who shared their expertise and insights on gene drives. Thank you for accepting our invitation, and we are truly honored to have you as our speakers for this webinar. To formally close our webinar, please welcome Mr. Neil Arman Padlan, Communications Officer at DABPO, who will read the message from Dr. Paul Limson, the Director of DA Biotech Program Office.
Good morning, everyone. On behalf of our dear Director, Dr. Paul Silimson, I would like to extend my heartfelt gratitude and appreciation to ISA Inc., led by Dr. Rodora Albimita, for inviting the DA Biotech program to this second part of the webinar series on gene drives. It is indeed a great honor and opportunity to learn more about the breakthroughs in gene drive in addressing some of the most pressing challenges in agriculture and environmental management. Throughout our discussions today, we have navigated the promising landscape of gene drive strategies. And by harnessing these advanced technologies, it offers us innovative solutions to lead more sustainable pest management techniques, reduce reliance on chemical pesticides, and the preservation of native species. These strategies not only aim to boost agricultural productivity, but also ensure the conservation of our natural ecosystems, working harmoniously towards the betterment of our sectors. To Dr. Paul Thomas and Dr. Maria Jeneline Diaz, they have illustrated how, through careful scientific advancement, gene drives can enhance food security and safeguard our biodiversity. We were honored to have them share their insights and expertise. With that, thank you, sir and ma'am, for sharing your insights and expertise. Thank you also to our esteemed speakers, dedicated panelists, and engaged participants who have made this event such a resounding success. If there's one message that the DA Biotech program wants to impart, it is always to carry forward the knowledge and experience gained to forge collaboration between researchers, policymakers, and communities. We need to harness our collective strength and share our expertise. This is the perfect time to unite our efforts and leverage these cutting-edge innovations for a sustainable future. Once again, thank you for joining us and contributing to this meaningful conversations. Thank you po.
Thank you very much, Sir Neil. Once again, thank you very much for everyone who joined us today. We hope to see you on the next part of this webinar series. Thank you, everyone, and have a great lunch.
Thank you so much, Professor Paul and Jenna. Dr. Jenna. Thank you.
Sure.
And Janine and Poppen and EJ and Clem and Tina. Sana sila? Yeah. Thank you so much.
Good. Lots of questions. Thanks.
Thank you. I think we'll have you again next year, Paul. Don't get tired of us.
I'll wait your email to come through, Ola.
All right. Thank you, Paul.
All right. See you all.
See you.
Bye.
Bye-bye. And also, Dr. Jenna, we'll call on you again in one of these webinars. We still have a lot
Yes po,
of things to
ma'am.
talk about. Thank you.
Okay po. Thank you. Bye-bye.
Bye-bye. Thank you.
Goody.
Good job. You too. You did a good job.
Goody, Janine.
Galing ni Jenny.
Galing ni Janela. Galing talaga ni Janela.
Oo, may premyo na yan.
Opo.
Sa Pasko.
Pasko. Nasaan na ba
Okay.
ako? Thank you very much, everyone.
Oh, thank you po, Ola Tita.
Oo.
Okay, kain na tayo.
Yeah. We will still give until 12 o'clock for others to complete the evaluation.
Oo. Okay.
Malaki
Good
dito
job.
natin at
Ang
147.
dami, 147. Oh,
Yeah,
pinakamataas.
that's the highest. That's the
Oo.
participants.
Mm-mm.
Okay na yun.
nga.
Marami interested
Mm-mm.
talaga sa G
And then,
Drive.
questions.
Questions, yeah, oh. Hindi ko na nga maano yung iba eh.
Oo.
There was one
Ito na
asking
talaga
about
interest.
application of the G Drive sa Monkey.
Oo nga. Ang daming monkey. Ay, sa ibang nations kasi marami
Oo.
talaga. Pero depende.
Ano talaga yun?
Ay, hindi. May research kasi naman doon. May ginagawang mga human-related research. Kaya,
Naging pest na ang monkey sa kanila eh.
sa kanilang countries.
Yeah.
And even, ano nga tawag dito?
Yeah.
Monkeys. Meron pang isa na...
Hindi ko ba is there in the... Parang ano to eh. Ang gawin kaya natin ay focus on invasive species. Biocontrol
Actually,
talaga.
meron eh. Actually,
Oo.
meron. Yung sa emerging ag.
The
Dati,
biocontrol.
invasive species talaga yung ano.
Yeah.
Kaya lang,
Ga-
yun nga yung tanong ko na nahuli.
Oh yeah.
Yung ano,
Hindi ko na nag-close na
kasi
ako.
yung,
Nag-cut
oo, ang tawag
na
dito.
ako ng...
Meron kasi, di ba pagka masyadong malalaki yung mammals na yung ganyan, matagal bago magkaroon ng ng gene drive
Yeah.
para ma-lead to extinction ng invasive species. Pero somehow organisms mutate para sila makasurvive. Yun yung one of the things na they have to look into, na dapat mas mabilis ang efficiency ng gene drive kaysa sa mutation drive. Mutation process. Kahit kasi anong species, nag-mutate siya eh. Ay ngayon may stress na siya in order to survive. Kailangan niya mag-mutate para siya yung mag-survive. Kasi meron ng gene drive. I think that's one of the things that we can discuss next.
Yep.
Okay, I have to go
Okay.
na ha. Nagtatawag
Okay,
na ng
sige po.
pagkain. Yung mga tao din eh. Bye-bye.
Okay,
Thank you so
bye-bye.
much.
Sige
Bye-bye.
po.
Congrats, Janine. And Poppen. Bye-bye na.
Thank you. Bye-bye. Can you picture AJ?
Okay.
That will be our last for the year.